Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 1 for January 2024. Let's begin with the first question. In question 1, red blood cells transport oxygen to the cells of the body. Read through the following description of how oxygen is transported from the lungs to respiring cells. Complete the description by writing the most appropriate word or words on the dotted lines. Oxygen binds to, this is hemoglobin inside red blood cells. Oxygen binds to this molecule in the lungs where the partial pressure of oxygen is high compared with elsewhere in the body. The oxygenated blood returns to the heart in the blood vessel called the pulmonary vein and is pumped to the body by the part of the heart called the left ventricle. In the tissues, the ability of this molecule to bind oxygen is affected by the higher concentration of carbon dioxide. This is called the Bohr effect. So this brings us to the end of question 1. Let's continue to question 2. In question 2, the nature of the genetic code of a gene is important in determining the sequence of amino acids in a polypeptide chain. The diagrams show the genetic code for some amino acids and the sequence of bases in part of a gene. So we can see this is part of the diagram given, this one here. Then down here they say part of a gene, which is this one here. The genetic code is described as a triplet code that is non-overlapping and is degenerate. Explain what each of these phrases means. They want you to use the information provided to illustrate your answer. So I said the triplet code, these are 3 bases that code for a single amino acid. For example, codon GCA codes for alanine. We can see all these amino acids are coded for by 3 bases, and that's what we call a triplet code. Then in part B, they say non-overlapping code. The code is non-overlapping because each base is read only once. A base from one codon does not become part of another codon. The codon AAA codes for lysine, while GCA codes for alanine. In part C, the genetic code is degenerate. This is because there is more information than required, and a single amino acid can be coded for by more than one codon. For example, both AAA and AAG code for lysine. If you look at all these amino acids, the first 2 letters or the first 2 bases are almost the same for a specific amino acid. For example, threonine is coded for by codons whose first 2 bases are AC. In here we can see proline, the first 2 bases are CC. So the information is almost coded in the first 2 bases. So it is a degenerate code because there is more information than required, and multiple codons can code for the same amino acid. So this brings us to the end of question 2. Let's continue to question 3. In question 3, the cell membrane determines which molecule can enter or leave a cell. Which pair of membrane transport mechanisms only involves a solute moving down a concentration gradient? This has to be diffusion and facilitated diffusion. Remember they said solute, so whichever has osmosis has to be cancelled out. And active transport involves moving against concentration gradient, so the answer should be a C. In part B, which of the following transport mechanisms involves carrier proteins in the membrane transport? Carrier proteins are involved in active transport, so the answer should be active transport only. And in part C, which cell transport mechanism is used to take large particles or bacteria into a cell? This is going to be endocytosis, so the answer is B. Which cell transport mechanism moves small lipid-soluble molecules through the membrane? Because they said small lipid-soluble, it's going to be diffusion. These are particles that are soluble in the cell membrane. And in part E, channel proteins are involved in facilitated diffusion. Which row of the table describes the charge on R groups on amino acids of the channel proteins? The first part says charge on R groups facing inside the channel are mostly, these are going to be polar. And then they say charge on R groups facing the rest of the membrane, these are going to be nonpolar because they're facing the phospholipids. So the answer should be B. And then part F, a student investigated the effect of 4 different colored solutions on the mass of pieces of sweet potato. Dyes were dissolved in water to produce the colored solutions. The diagram shows the method used by the student. So they say 4 pieces of sweet potato were cut and weighed, and each piece of sweet potato was placed in a different colored solution and left for 1 hour. Then each piece of sweet potato was removed, dabbed dry and reweighed. Then they say the graph shows the results of this investigation. On the vertical axis, we have the mass of the sweet potatoes, and on the horizontal axis, we have the colored solutions. We can see this is the initial mass and this is the final mass of the sweet potatoes. So for the sweet potato put in the purple solution, its mass was higher before, but there was a slight decrease. The sweet potato in the orange solution, there was a greater decrease in mass. This is because the solution was hypertonic, so a lot of water was lost. The mass of the one placed in the red solution increased. This means the solution was hypotonic, so water entered into the sweet potatoes by osmosis, increasing the mass of the potato. And for this one, there was no change. That means the solution was isotonic. This is solution blue. So there was no change in the mass of the potato because there was no net movement of water. They say, explain the results of this investigation. Like I've already explained, I say the mass of the sweet potatoes changes because water moves by osmosis. The mass of the potatoes in the purple or orange solutions decreased because water was lost by osmosis. The orange and purple solutions are hypertonic compared to the cytoplasm. Greater decrease occurred with the orange solution. The mass of the potatoes in the red solution increased. The red solution was hypertonic compared to the cytoplasm, so water entered the potatoes. The potatoes in the blue solution had no change in mass because the solution was isotonic compared to the cytoplasm, so no net movement of water occurred. So this brings us to the end of question 3. Let's continue to question 4. Water is an important molecule in living organisms. Water is a dipolar molecule that forms hydrogen bonds with other water molecules. Explain the dipolar nature of water. If we look at this water molecule, these bonds are polar. The oxygen-hydrogen bonds are polar. This means hydrogen is somehow positive in comparison to the oxygen, which is partially negative. So these are the ones showing the polarities. So I said in a water molecule, oxygen is slightly negative and hydrogen is slightly positive, or the hydrogens are slightly positive. There is an uneven distribution of charge on the molecule leading to polarity. The next I say the diagram shows 3 water molecules. Complete the diagram to show a hydrogen bond between 2 water molecules. You could just show a hydrogen bond, just one, between 2 water molecules. So if you use this one, the hydrogen bond should be between hydrogen and oxygen. If you want, you can even position the polarities, this being partially positive, like I represented them here, and the oxygen of this one being partially negative, so that the hydrogen bond forms between the hydrogen of one water molecule and the oxygen of the other. Also, this is a hydrogen bond, if you could draw it, but this angle does not orient properly. So I can just eliminate this. Let me eliminate that. So I have left only this one. This is better oriented in comparison to the other. In part B, water plays a role as a solvent. Sodium chloride dissolves in water. The diagram shows a sodium ion. Complete the diagram to show the arrangement of 2 water molecules around a sodium ion when it is dissolved. Here I use 2 water molecules. When sodium ion is dissolved, because it's positively charged, the oxygens of the water molecules orient themselves closer to the positive sodium, and then the hydrogens are going to be oriented away in this manner. In part C, temperature affects the solubility of chemicals in water. The graph shows the effect of temperature on the solubility of 4 chemicals. We have chemical A, B, C, and D. On the vertical axis, we have the solubility of the chemical, and on the horizontal axis, we have temperature. We can see for some of these chemicals, as the temperature increases, solubility increases. For example, for chemical A, there is greater increase in solubility. Actually, the relationship is linear. But for this one, we see an exponential increase and then decrease. This one looks like it has a very slight increase, and there is a decrease in solubility of chemical D as the temperature increases. So they say describe the effects of temperature on the solubility of these chemicals. Like I've explained while looking at the graph, we saw for A, an increase in temperature leads to a linear increase in solubility, and for B, an increase in temperature leads to an exponential increase in solubility until 32 degrees Celsius. Then the solubility decreases as temperature is increased. And for C, initially, an increase in temperature had no effect on solubility until about 20 degrees Celsius. However, after 20 degrees Celsius, increase in temperature leads to a slight increase in solubility. And for D, increase in temperature decreases the solubility. So this brings us to the end of question 4. Let's continue to question 5. In question 5, several factors are associated with the risk of developing cardiovascular disease. Obesity is one risk factor associated with CVD. The ability of a group of people to lose weight was investigated. This investigation considered the body mass index of a person, the level of amylase in their saliva, and the bacteria present in their digestive system. The BMI of a person can be calculated using the formula BMI is equal to mass in kilograms divided by the height in meters squared. They say name one other obesity indicator. I wrote the waist to hip ratio. Then the next part says which of the following is the BMI of a person who has a mass of 65 kilograms and the height of 165 centimeters. Using this formula here to calculate the BMI, the mass in kilograms is going to be 65, we have it here, and the height is 165 centimeters, which is 1.65 meters. So BMI will be 65 divided by 1.65 squared, giving us the answer 24, and B is going to be the answer. The next part says a person's BMI is used to put them into a weight category. A person with a BMI of 25 and above, but below 30 is considered to be overweight, but not obese, which describes the body mass of a person who is obese. Based on this, an obese person is going to have a BMI greater or equal to 30, so the answer should be B. And in part B, people who are overweight or obese are encouraged to lose weight. State 2 ways in which a person could lose weight. They could lose weight by doing more exercise, as well as eating food that contains less calories or decreasing their calorie intake. In part C, amylase in saliva breaks down amylose into maltose. Which row of the table describes the action of amylase? Because they said amylose and amylose contains only 1,4 glycosidic bonds, the type of reaction is going to be hydrolysis, and the glycosidic bonds broken are going to be 1,4, so the answer should be a C. Then next they say, people with low levels of amylase in their saliva were found to lose weight more easily. Suggest why these people lost weight more easily. If they have low levels of amylase, then it means starch is not going to be fully digested into the monosaccharides, for example, glucose, and so their body will not take in more glucose, so they'll lose weight easily. So I said having low levels of amylase leads to less starch digestion into maltose, so less glucose will be formed and absorbed in the body. Less glucose will be stored as fat. Also, more fat will be respired to provide energy. In part D, the bacteria in people's digestive systems replicate. The investigation found that people with greater weight loss had bacteria with increased rates of replication. Explain why this finding can be described as a positive correlation. It's a correlation because as bacteria replication rates changed, weight loss also changed, and the correlation is positive because as replication increases, weight loss also increased. Then they say suggest why the rate of replication of bacteria could affect weight loss. Bacteria are going to carry out respiration using the food that is broken down, so less glucose is going to be absorbed into the body. So here I said because energy is required for bacterial replication, and this energy is generated from respiration of digested food. So if this occurs and the bacteria are many in number, less glucose or fats are absorbed into the body, leading to weight loss. So this brings us to the end of question 5. Let's continue to question 6. Question 6. The cell membrane consists of phospholipids and proteins. Name the current model of the structure of the cell membrane. This is the fluid mosaic model. And in part B, they say describe the arrangement of phospholipids in the cell membrane in this model. In this model, the phospholipids are arranged in a bilayer. The hydrophilic phosphate heads face outwards, of course towards the aqueous medium, and the hydrophobic tails face away from the aqueous layer towards each other. In part C, phospholipids can be used to form lipid nanoparticles. Lipid nanoparticles can be used to carry nucleic acids, which could be DNA or RNA, to tissues in the body. The diagram shows a lipid nanoparticle containing messenger RNA molecules. We can see the hydrophobic tails are facing towards each other and the hydrophilic heads are facing away towards the aqueous layer. Also on the other side, we see the heads are facing towards the messenger RNA as the tails face away. Next, they say lipid nanoparticles are very small. Calculate the diameter of a lipid nanoparticle with a volume of 0.108 micrometer cubed. They want us to use this formula, and pi is equal to 3. So if volume is 4 over 3 pi r cubed is going to equal to 3 times volume divided by 4 pi. And then substituting in the volume, r is going to be the cube root of 3 times 0.108 divided by 4 times 3, giving us 0.3. Now this is going to be the radius, but the question wants us to find the diameter. And we know diameter is twice the radius, so it's going to be 0.6 micrometer as the answer. Next, they say describe the structure of a messenger RNA molecule. Messenger RNA is going to be single-stranded. It has a phosphate sugar backbone. It's made up of ribose sugar. It contains bases adenine, uracil, guanine, cytosine. And it contains phosphodiester bonds between the mononucleotides. So any 3 of those, you get full marks. Next, they say explain the arrangements of phospholipids in this lipid nanoparticle that carries messenger RNA to tissues in the body. I have actually gone through this with you when we were looking at the arrangement of the lipid nanoparticle. So the phospholipids contain hydrophilic heads and hydrophobic tails. The hydrophobic tails were facing away from the aqueous environment, and we saw that some tails were facing each other. The inner phospholipids surround the messenger RNA using their polar heads. So messenger RNA is polar due to the phosphate groups present on the backbone. So this brings us to the end of question 6. Let's continue to question 7. In question 7, cystic fibrosis is an inherited disorder caused by a mutation in the gene coding for the CFTR protein. Explain how this mutation results in reduced gas exchange in the lungs. When this mutation occurs, the CFTR protein produced is going to be non-functional and it will not be able to cause the movement of the chloride ions from the cell into the outside, so the mucus is not going to be diluted, and then we're going to form thick and sticky mucus, which is going to affect in multiple ways. So I said a mutation leads to a non-functional CFTR protein, so chloride ions won't be transported outside the cell, and due to that, water will not leave the cell to dilute the mucus, so the mucus will become thick and sticky, and the cilia will not be able to move the mucus properly, so the mucus will block the pathway for air, decreasing the surface area for gaseous exchange. And this question was in reference to reduced gaseous exchange in the lungs. Next, they say the number of new cases of cystic fibrosis in USA in 2015 was 964. It is predicted that this value will have fallen to 839 by 2025. Calculate the predicted percentage decrease in the number of new cases from 2015 to 2025. The decrease is going to be 964 minus 839, giving us 125. So 125 divided by the original, which is 964, times 100 gives us 12.966805, which is about 13.0 or 13%, and that is the percentage decrease. Next, they say explain why the number of cases could fall. This is easy. People are more aware, there is more genetic testing, and so people are choosing alternatives instead of having children. Maybe they choose to adopt or they go through IVF. So I say there is more genetic screening. They can do pre-implantation or prenatal, and more couples can choose to use IVF, in which only normal embryos are implanted. There is also more awareness, so more carriers are identified, and some couples make a choice not to have kids if they are carriers. The allele has no genetic advantage, so its frequency is decreasing. There is also this possibility. One complication of cystic fibrosis is lung disease caused by the buildup of bacteria in the airways. This may be due to the inability of the person to clear mucus from the airways. People with cystic fibrosis can inhale a salt solution to treat their symptoms. This gives improvements in the clearance of mucus, lung function, and quality of life for a short time. It has been suggested that pretreatment with drug A could extend the effects of inhalation of a salt solution. Devise an investigation to determine if pretreatment with drug A is more effective than inhalation of a salt solution on its own. Give reasons for your choice of method. So here we're going to design an investigation, and we will have two groups of people. To one group, we'll first give drug A before giving the salt solution, and to the other group, we'll just give the salt solution. We're going to make sure that it's double-blind, and we use large numbers of people so that the experiment or the study can be valid. In doing so, we have to use people of the same gender, same age, and they should have similar health risks or similar cases of cystic fibrosis, and they should not be taking any other medication, or they should be taking the same type of medication. So I say set up two groups of people. To one group, give only the salt solution. To another group, give drug A before the inhalation of the salt solution. In testing, the people should be the same age, gender, and with similar issues related with cystic fibrosis. The volume and concentration of the salt solution should be the same for all participants. All recipients of drug A should receive it in the same form, meaning it could be tablet or it could be liquid. It has to be the same form and concentration, and participants should not be on other medications. The trial should be double-blind to avoid bias, and the participants should be followed for some time to ensure consistency in the results. This could be maybe 6 months and so on. They should use a spirometer to measure the changes in lung function, so they can use the spirometer before and then after the study to see if there are some changes as time progresses. And then they can use a suitable statistical test in order to know whether taking solution A was advantageous. So this brings us to the end of question 7. Let's continue to question 8. The types of lipid consumed in the diet can affect the health of individuals. Tropical sprue is a disorder that limits the absorption of nutrients into the blood. Consumption of unsaturated lipids has been proposed as a cause of tropical sprue. The diagram shows part of an unsaturated lipid. Complete the diagram to show one possible structure of a fatty acid side chain of an unsaturated lipid. Because they said unsaturated lipid, and we're talking about the fatty acid side chain, you can draw at least one carbon-carbon double bond. Of course, it shouldn't be this one. It should be either here or the other carbons. And then fill out with hydrogen, since this side should be only carbon and hydrogen. So a structure similar to this, with all the hydrogens displayed, would be okay. In part B, consumption of high levels of cholesterol is associated with atherosclerosis. In the development of atherosclerosis, lesions form in the coronary arteries, and these develop into atheromas, which are plaques. In an investigation, one group of primates was fed a diet supplemented with cholesterol and saturated fatty acids. A second group was fed a diet supplemented with cholesterol and unsaturated fatty acids. The primates were fed this diet from a young age and then monitored over a period of 60 months. It's kind of a long study, though not too long. Then the primates are a higher order of mammals that include monkeys. They say the results of the investigation are shown. So we can see from the key, these are for saturated lipids and these are unsaturated lipids. And for the first graph, on the vertical axis, we have the percentage of surface of arteries covered with lesions as time passes, all the way to 60 months. We see there are more percentage of lesions with those who took saturated lipids than those that did not. We can see it's increasing over time. There are more lesions with saturated fatty acid consumers. And in graph 2, on the vertical axis, we have percentage of primates with atherosclerosis. And of course, we see time on the horizontal axis. And even here we see there are higher percentages among those who consumed saturated lipids in comparison to those who took unsaturated lipids. In graph 3, we see the mean area of atheroma after 60 months. Again, here for saturated lipids and unsaturated lipids, we see there is greater mean area of atheroma for those that took saturated lipids, but it's way higher in males. Here we can see it males, but they are not females. So this tells us these results are gender related. There is connection with gender. And because we see the error bars are put, the mean values were used, so it tells us the experiments were repeated, the mean were calculated, and then error bars were included. There is no overlap between males and females that took saturated lipids. However, there is overlap between males and females, meaning for females that took saturated and males that did not. So the difference between these results is not significant, where the difference between these would be significant. So they say calculate the mean rate of increase in the surface of arteries covered in lesions from 16 to 60 months in primates given saturated lipids. This is from graph 1. In graph 1, we can see here we have 2, and here we have 52. So the difference is going to be 52 minus 2, which gives us 50, and then divide by the difference in the time, which is 60 minus 16, and the answer was 1.14 percentage per month. And next they say calculate the ratio of primates with atherosclerosis given saturated lipids to those given unsaturated lipids at 32 months. This is from graph 2. From graph 2, we have at 32 months, we have this is going to be 32 and this is 22. If you can look at the vertical scale clearly. So in our calculation here, the ratio is going to be 32 to 22, which is 1.45 to 1, rounding off to 1.5 to 1 as the ratio. Next, they say calculate the difference in area of atheromas in males compared with females in primates given saturated fats from graph 3. From graph 3, we have 0.026, if you can calculate clearly, and this is 0.004. I have the difference 0.026 minus 0.004, giving me 0.022, but this is millimeter squared. I need the answer in micrometer squared, so 1 millimeter is 10 power 3 micrometer, and 1 millimeter squared is going to be 10 power 6 micrometer squared. So this in millimeter squared, we'll multiply that times 10 power 6, giving us 22,000 micrometer squared as the answer here. Then down here they say, these primates were fed a diet supplemented with cholesterol and either saturated lipids or unsaturated lipids. Deduce the effects of these two diets on the development of lesions in the coronary arteries, atherosclerosis, and atheromas. To answer this question, I'm just going to go through one graph at a time. I'm just going to read what I wrote because I've already explained the results when we're going through the graphs. So from graph 1, the percentage of surface area of arteries covered with lesions increases for both diets over time. However, there is greater increase among primates fed on saturated lipids than unsaturated lipids. This shows that the type of diet can have an effect on formation of lesions within arteries. However, in that experiment, there were no repeat experiments, there were no error bars, so the results may not be valid. From graph 2, the longer primates feed or fed on both lipid diets, the greater the percentage of primates with atherosclerosis. There is higher percentage with saturated lipids, however, no repeat experiments, and there are no error bars, so the results may not be valid. And from graph 3, there is greater mean area of atheroma in primates that consume saturated lipids and those that consumed unsaturated lipids. Males had greater mean area of atheroma than females for both types of lipid diets. Females with unsaturated lipids had 0 millimeter squared area of atheroma, so gender influences atheroma formation. From this graph, means were calculated, error bars were included. However, overlap exists between results for females on saturated lipids and males on unsaturated lipids, so the results are not valid. And finally, they say discuss the ethical issues relating to use of primates and other animals in investigations such as this one. In questions about discussing, we have to talk about the pros and cons. So I say the animals may die from complications relating to heart disease. Also, the animals do not consent, to participate in these kinds of studies, so it's kind of unfair. However, we look at the benefits. Because primates are closely related to humans, the results from the studies are helpful in finding ways to combat CVD or to find how CVD progresses, and this is helpful to the humans. Also, using the animals is more ethical than testing on humans. So this brings us to the end of question 8 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.